So this is going to be talking about Aplysia californica as a model for synaptic plasticity. And this is the invertebrate model, and I'll talk about the mammalian model in the next video. But this is a way to introduce the concepts of plasticity. The concepts of facilitation, I can't spell today, facilitation, which is the short-term strengthening of the synapse. Short-term strengthening of synapse. Kind of using shorthand here. Depression, or the opposite of facilitation. Can't remember if there's another word for it, but this is going to be the short-term weakening of a synapse. So the difference between these two ideas and then this long-term potentiation. So long-term potentiation, which is, again, a long-term strengthening of a synapse, whereas long-term depression would be a long-term weakening of a synapse. And we'll talk about these in more detail as we go through this. But first, what I want to talk about is the fact that facilitation is also sometimes called habituation. In fact, the term habituation can actually mean both short and long term, so we're going to kind of avoid that term. In addition, some people say like sensitization for depression, which is the fact that these neurons will stop responding to a stimulus. So we're just going to go with depression and leave it at that, but just know that other words exist out there. So what leads to these short-term changes? Well, we know that when we fire an action potential down the neuron, once it gets to the terminal, we end up having calcium influx. But we know with equilibrium potential that eventually that calcium, after it's kind of used for what it does, it'll kind of leave out of those calcium voltage-gated channels to restore that potential, right? You have an equilibrium potential for calcium that wants to be met. And you'll have these channels that kind of pump calcium out to maintain that. But let's say you fire two synapses or two action potentials in close sequence. So you fire one, and let's say you get 3 molecules of calcium in, right? And of course, in reality, it's going to be a lot more. But let's say you get 3 molecules of calcium in. So action potential 1, get 3 molecules of calcium. But then, the action potential fires, you get neurotransmitter release, and then calcium starts to leave. So you get calcium leaving. But in very close sequence, you have another action potential. So this is action potential 2, and there's a short delay between these two. So what you would get is buildup of calcium in this synaptic terminal, and what that would lead to is even more neurotransmitter release in response to an action potential. So the response of the second action potential is actually larger than the first action potential, and that's because of this buildup of calcium, and it's also due to a couple other factors. These are just examples I'm using of facilitation and depression. Another thing that could happen is depression, right? So if I erase this and we think about more like the kinetics of a channel, as you fire an action potential, calcium channels open, but eventually they're going to close, right? So if this is your calcium channel and it lets in a certain amount of calcium, but then it closes, it inactivates, there's a short period of time, there's a time delay in which you can't open those channels again. They need to get back to a steady state. And so what ends up happening is that because these channels are closed, you get short-term depression, which is the short-term weakening of a synapse thanks to this phenomenon. In addition, and I'm sorry I'm going really fast, but you're welcome to pause all of this. I'm just trying to hit that 15-minute mark. But in addition, what can happen is all those vesicles in your readily releasable pool can be exocytosed so quickly that you can't really replace them in that short amount of time. And so let's say you fire action potentials so quickly that all of your readily releasable pool is expended, and then you have to keep going and keep, binding those snare complexes, and you can't do it fast enough to allow for those vesicles to get in that readily releasable state. That could also lead to short-term depression. And I cover this in my exam review sheet too, so you guys can take a look at that there. Because I kind of want to move on to the model organism we talk about. So the sea slug Aplysia californica has a couple prominent structural features that I want to talk about. So this is known as the siphon. The siphon contains a sensory neuron. So that sensory neuron I want to call sensory neuron 1. So this is a sensory neuron, has dendrites. It responds to a stimulus that we'll talk about in a sec. But this is sensory neuron 1. And it is conjugated to a motor neuron that affects this muscle right here. So this muscle, underneath this region, this is going to be the gill. This muscle is the gill withdrawal muscle. And then here's your tail of the sea slug, and here's your head. So here's your head and your tail. So the reason they have this synapse, or this neuron that connects to this motor neuron, I identified this as the motor neuron, is because in response to water, excessive amount of water flowing over this animal, this sea slug is going to want to contract this gill withdrawal muscle to prevent the gill from getting too much water in it, which would damage the gill itself. So this sensory neuron actually responds to water. So if too much water, if you were to shoot water out of a little pipette at it, it would contract like this. So if there's too much water, the neuron fires onto the motor neuron and the muscle contracts. Muscle contracts. So they found this and they described this as a monosynaptic circuit. You get a sensation immediately conjugated to a action, right? So this is just a basic reflex arc. But what they found is that if they shock the tail, so if they deliver quite a substantial electric shock to the tail, the next time they shot water onto this siphon sensory neuron, the gill withdrawal reflex would actually be bigger. So I can write that down. After tail shock, response larger, or I'll say gill withdrawal reflex larger. Reflex are greater. So what they found, because of these studies, this is Eric Kandel, he's the guy who we showed a video of in class, there's actually a second sensory neuron here in the tail. And what that projects to is an interneuron just being something that connects to a circuit and kind of modulates its activity. So they found and they hypothesized that something about this interneuron facilitated the response of this sensory motor integration circuit. So something about this interneuron, this is the L29 interneuron, makes it easier for this sensory neuron to send information to the motor neuron and cause it to contract. So they didn't know what it was that did this. They didn't know if it was making the terminal of the sensory neuron more easily or more readily releasing neurotransmitter, or if the motor neuron was being the receptors on the motor neuron were being upregulated, or something like that was facilitating this response. So what they did when they, got a couple better techniques in neuroscience for studying this kind of stuff, they found that they can actually describe exactly what's going on here. So if this is your L29 interneuron, they discovered, this is L29, that it was serotonergic, meaning it releases serotonin. And they found that it was actually adhered to the sensory neuron. So it's not the motor neuron we're talking about here, it's the sensory neuron. And then here would be your motor neuron. So something about this L29 neuron is causing an increase in the excitability of the synaptic terminal of the sensory neuron, right? So this is the sensory neuron. So what they found is that this sensory neuron contains a G protein coupled receptor conjugated to a G alpha S. So that's a stimulatory alpha subunit of the G protein, heterotrimeric G protein complex. Right, and this serotonergic neuron is releasing serotonin. This GPCR has a specificity for serotonin. So this is probably like a 5-HT something. Not a 5-HT3 though, because 5-HT3 is inotropic. So what happens, and you guys remember from the G alpha S pathway, that this eventually leads to protein kinase A activity, right? So we get this positive effect on protein kinase A. And the effect of protein kinase A in this case is to do two things. The first thing it does is it inhibits potassium channels. So this is a potassium channel. It might be a GERK channel, for example. What it does is it inhibits this channel so that potassium remains in the cell. So it remains in the presynaptic terminal. This increases the excitability of this terminal because more positive is maintained within this presynaptic terminal. In addition, protein kinase A also upregulates the activity of a voltage-gated calcium channel, or a CAV channel. And this leads to more calcium influx, more neurotransmitter release. So what's happening here is that by shocking the tail, you're increasing the activity of the serotonergic L29 neuron. And the effect of that is to increase the excitability of this presynaptic terminal of this blue sensory neuron so that it's releasing more neurotransmitter, it's more excitable, and this response of the gill withdrawal reflex is larger, right? And this is most likely an evolutionary response to some kind of danger, some kind of predator. It wants to protect all the vital organs that exist within this gill area. That's probably where maybe the heart is, or I'm not exactly sure. But basically, what you have is a serotonin-induced increase in synaptic transmission thanks to this circuit. In addition, PKA, if I were to talk about PKA a little more, so cyclic AMP, for example, we've talked about the fact that increases the activity of PKA. What it does is PKA is actually inhibited by these regulatory subunits. So this is a regulatory subunit. And PKA is turned on, quote unquote, by cyclic AMP inhibiting the regulatory subunit. So what happens, and I can draw this out as a kind of disinhibitory circuit, but PKA activity is inhibited by the regulatory subunit, so I'll call this reg, but cyclic AMP disinhibits PKA. So what it does is it removes the inhibition, leading to an increase in PKA activity. So the reason this is important is because cyclic AMP also has another function. So when it's produced as a response to, so this is going to be, I simplified this a little here, but this is going to be cyclic AMP activity, and that's going to lead to PKA activity. In addition to this more short-term facilitation, so this is short-term, you also get cyclic AMP affecting transcription. So this increases or upregulates a transcription factor, which encodes for a protein protein regulator. And the effect of this protein regulator is actually to also inhibit these regulatory subunits. But whereas this cyclic AMP is more transient as a result of serotonin binding to this GPCR and upregulating the production of cyclic AMP, this protein regulator is going to be more affecting baseline PKA levels because this protein regulator is going to be transcribed by the DNA. So this is always in the neuron, right? So this is always working to increase the excitability of PKA, always working to increase the amount of activity going on in this protein kinase A, which remember has a lot of functions, one being to increase the excitability of this excitable or this excitatory blue sensory neuron. And if I was to talk about the neurotransmitters here, I know I mentioned serotonin is released by this one, right? And if I was to redraw this, now that we know what we know, I would draw it interacting with the terminal of the sensory neuron. So we know the green one is serotonergic, the blue neuron is actually glutamatergic, and then the red motor neuron is going to be cholinergic or acetylcholine. So it's going to release ACH. And yeah, that's it for this video. Thanks.